wave experiment. I'll be planning. The one I want to have us do is thermal camera.
Okay, I think we're gonna get started here.
This week.
And so I'm Jen Mixes-Olds, and I'm here at UNH. UNH is doing the web hosting, and Brown University this semester has organized all of the speakers for our Northeast Regional Environmental Acoustics Seminar. And we have 2 speakers today. But before we start with speakers, I just wanted to remind everybody that there is a website that tells you when all the upcoming events are. So we have got 2 speakers today, which I'll introduce in just a minute.
That's funny.
I'm going to ask everyone who's not speaking to mute so we can have, there we go, that's better. I'm going to turn my email off so you guys don't get to read all my email while I'm, let's see here. If I can get my email off, there we go. Next month, we also have 2 speakers, and that's going to be all about bats. So that's on April 16th. And then I wanted to actually have everybody here mark their calendars, May 29th at Brown. We've got 2 keynote speakers. One is listed here, Holger. The other is Jesse Barber from the National Parks. So we'll have 2 very awesome, engaging keynote speakers. So put this on your agenda, and then within the next week or 2, you'll be able to come back to this site, and there'll be a link to submit abstracts if you want to give a talk or a poster. And so that will be coming up in the next week or so. So with that, I am going to stop sharing and allow the Cornell group to start sharing. And while you start sharing, I just want to remind anybody in the chat area, it would be great if at some point you could just send me a chat telling me how many people are viewing with you, because we like to just keep track for our own record keeping. And so that would be very helpful. So we have 2 speakers, both postdocs at Cornell. Our first speaker is Dr. see if I get this right, Shyam Madhusudhana. I'm sorry, I butchered that. I apologize. Shyam has a bachelor's in computer engineering, a master's in computer science, and a PhD in applied physics. His research interests have been largely multidisciplinary, with applications in autonomous monitoring and conservation of marine and terrestrial fauna. As a postdoc at Cornell, his research involves developing solutions for automatic source separation in continuous ambient audio streams and the development of deep learning techniques for unsupervised multi-class classification of acoustic events in the big data realm. So at this point, I'm just going to turn this right over to Cornell and let you guys take it away. We're going to do about 20 to 25 minutes for each presentation and allow about 5 minutes of questions in between, if that's okay. Oh, there we go. I don't hear anything coming from the Cornell side. Are you muted?
Can you hear us?
Now I can, yes.
Yeah, thanks for the introductions. Yes, so as the title says...
You're a little bit quiet. I don't know if you could get closer to your speaker.
Is this better now?
No.
How about now?
I think just go, I think, with the best that you've got, and we'll make it work over here.
Okay, sorry about that. Yes, as Jen said, I've worked with multiple realms, both terrestrial and acoustics. And although the title of this presentation says bird call classifier, the techniques proposed here are applicable to other domains as well. So there's ongoing projects where we're working on...
This is Jim at Brown. You need to make it louder somehow.
Does it sound any better? Hello?
Computer you're using.
Can you hear us now?
We could hear you. You're just very quiet, like you're far away from your mic.
I'm actually right on the microphone.
That's better.
Actually, is it better now?
Oh yeah, much better.
All right, I'll continue with this. So yeah, as I was saying, all the title says bird call classifier. All of these techniques that I'm going to present, that's applicable to other realms as well, including marine sounds. Just to talk a little bit about artificial neural networks. Basically, it's basically mimicking neural networks as it happens with biological organisms. You have your input nodes, synapses, activations, and the output nodes, and the information flows through. When you have multiple hidden nodes, that becomes deep neural networks. Um, the most common form of neural network that's being used in the industry today is the convolutional neural networks, which is more suited for multi-dimensional inputs. Basically, what you do is you take your input, you apply convolutional filter operations. That gives you an internal representation of the input image, and then you pool them. Basically, pooling is reducing spatial dimensions. So you're actually compressing the images, and then you apply further filters, you increase the width, as in, increase the number of channels that you have, increasing the number of representations of the input images subsequently, and then you pool, and then you convolve again, and so on, as long as it's desirable. And then subsequently you have your fully connected layer where it merges all of the previous layers, and then you have your output layers. You can look at this as if, if you were doing facial recognition, the initial convolution layers would highlight, low-level image features like edges and gradients, and the subsequent layers would identify more domain features like edges of faces or eyes or nose, and then the further subsequent layers would identify relationships between them, and the fully connected layer eventually stitches them together to be able to classify something. So one of the major application areas for convolutional neural networks is computer vision. It's one of the most widely researched areas today. Almost all of the big software companies, the data science companies, are fully devoted to research in this field. Name it Microsoft, Facebook, Google, everybody's into it. Of course, you have a lot of applications for this technology. Again, face detection, person detection, species classification, license plate recognition, name it, you have every application in this domain. Most of them can be categorized into either object detection or classification. With our problem of using time domain signals, that is the acoustic signals, we always represent, most of the analysts looking at this domain either look at spectrograms or MEL spectrograms or MFCC, something like that, to be able to analyze them. So going by that route, if we use convolutional neural networks, what we essentially want to do is replace those photos with spectrograms, basically. So that's about it. That's how most acoustics-based species recognition happens in today's world. There was a open bird acoustic detection challenge in late 2016, early 2017, organized by university in the UK. And according to them, most of the submissions, which was about 30 plus submissions, all of them were based on CNN-based techniques, and all of them use MEL spectrograms as inputs. That's just to show how popular these techniques have been. So there's, with convolutional neural networks, there's multiple types of architectures that you can come across. Shown here are some of the examples. Mind you, all of these are truncated, so they, the full network is not shown. Just to highlight I'm just highlighting the early layers of these networks. The common feature of all of these networks is the convolutional layer, the initial convolutional layer, which is also sometimes known as the preconv. Basically, it's meant to highlight, again, very low level features in spectrograms or images, as in edges and intensity gradients. Because we're dealing with spectrograms, those are not the spectrographic features that we want to be highlighted. So there's obviously scope for improvement here, where we can add in a front end or a pre-processing layer that fits in before the initial convolution layer, thereby enhancing the features that we want to be enhanced. Or we can have another route where we have a drop-in replacement for the convolution layer. One of the popular methods that's being used currently in the industry is the PCEN, which stands for per channel energy normalization, which is basically the equation on the right of the screen here. You basically apply that equation along each frequency bin of the spectrogram. What it does is enhances the onset of speech, which is vital for applications such as the hot word recognition. When you say, hey Siri, hey Google, things like that. So it's vital for the onset of the spoken signal to be recognized there. Yeah, the researchers showed this to be much better, helping much better than regular spectrograms or MEL spectrograms. The downside of this equation is that there are 5 parameters, and this, it really doesn't, it's not intuitive to choose values for these parameters. And then, even slight changes to any of these parameters has unexplainable changes in the output, because it's more like an IIR filter. And needless to say, the outputs of applying this method have no bearing with physical measurements. So you don't have any idea of what the output relates to in terms of the signal to noise ratio, the actual pressure level, things like that. One way that the authors of this paper proposed to set the parameter values is to actually fit this module into a convolutional network and have the system learn the values for the parameters. This is advantageous where you don't have to worry too much about where to start with. You just set it with some random values, and then the system learns the best representation. And yet again, those parameters will be more suited to a particular domain. So the authors also suggest that we use a suite of these PCN modules, all trained with different weights, which will eventually feed in as an aggregate to the subsequent network. Now, because we're dealing with spectrograms, it's obviously it's very different from images. Any signal that a human being can pick out in spectrograms or in any other type of time frequency representation is signal that stands out in relation to its tempospectral vicinity. So all of these signals that are readily detectable fall in a time frequency continuum where signals that are spread across a number of frequency bins are considered broadband signals. Signals that have very small distribution across frequency are called tonal signals or narrowband signals. So our approach is to basically build a system that highlights these features rather than edges and gradients. So specifically, we work with intensity differentials. In other words, we actually consider second order differentials. So the approach proposed is a deterministic closed form approach. So there's no learning involved, and the system behaves the same way no matter what kind of inputs it is. And we propose this to be a drop-in replacement to the pre-conv layer instead of an additional layer. Just to go over how we do feature extraction without going into too much details. Shown here are examples of plots taken from the original papers published in the 90s. The plots on the left correspond to blob detection. The ones on the right correspond to ridge detection. In each pair, the one on the left is the original image after application of a spatial smoothing filter. In other words, it's a Gaussian blurring filter. So you'll see that Gaussian filtering is applied at different scales, and each of the, and the right ones in each pair is the output of applying the mathematical computations where you pick out the ridges or the blobs. You can see that at different scales, different regions of similar intensities are picked out in the blob detection. In the case of the ridges, there's the outputs contain black lines. You can see that at very fine scale, there's a lot of ridges detected because of the noise. But as the scale gets coarser, you can see, the central line along where the hand is, that's actually an intensity ridge, which is, picked out really nicely. So we use these techniques with little modifications so that they actually work well with spectrograms. Here is an example of application of these techniques to spectrograms. The top row is what most people are comfortable with, the jet color scale. The bottom ones have the outputs, but then they're all normalized, so they're in the range 0 to 1. Anything that's 0 is white. Anything that's the darker ones indicate that the values are close to 1. The top two the top two scales in the output are well suited for detecting ridges, which is tonals or whistles. The bottom 3 in this case are well suited for broadband signals. You can see that some closely spaced tonals also appear as broadband signals in the bottom 3 plots, but then the system will be capable of learning these through. One thing to note here is that the regions of the spectrogram that do not seem to contain signals, detectable signals, they're all set to 0. Going forward, just to be able to compare our proposed method to what's being used currently, I've shown here output of the pre-con filter for one of the models that was already trained. So this is a model with 16 pre-con layers. You can see that the layers have actually done a good job here. Some of them have picked out the vertical edges. Some of them have picked out horizontal edges. Some have picked out the oblique edges. Some have picked out the positive edges. Some have picked out negative edges. The thing to consider here is that the range of values in each of these outputs, some are predominantly negative, some are predominantly positive. So theoretically, it makes it harder for the system to learn something from this. In comparison to our method, you can see that the values are well normalized. So the subsequent batch normalization operations in typical convolutional networks can actually benefit from this. Yeah, as opposed to PCEN or the pre-con method, there's better relationship with the output of our method, the actual signal to noise ratios of the underlying signals. And one thing to note here is that the regions of spectrograms that do not seem to have any signal are all set to 0. As a side note to this, there was an experiment where again with the cats and dogs experiment, researchers trained a system with images of cats and dogs, but then they had all the cat images with red background, all the dog images with green backgrounds. And when you run the system with images which have these colors swapped, the system performed badly, which goes to indicate that the system actually did not learn the features of cats and dogs, but actually learned to recognize the background. So that situations like that can be avoided with this system. Well, yeah, that goes along notion of domain adaptation, which is well researched in this field these days, wherein you train your system with inputs from a particular region, but then the system doesn't perform well when you have inputs from a different recording environment. So this system helps avoid such situations. What's shown here is the results of applying same network with 3 different types of convolutional filters, and then the last column shows the output wherein the intensity differentials module was a drop-in replacement for these filters. As you can see, the number of channels that goes into the subsequent layers decreases in the intensity differentials. This means that it has a lot less to learn from, although it retains all the information. The number of trainable parameters does show decrease as we go down to the end of the last column. Not to read too much into these numbers because it's a very simplistic model, but as we deal with complex inputs, a lot more species, a lot more environmental data, the network needs to be a lot more complex, in which case the number of trainable parameters increases exponentially. This can be avoided by using intensity differentials. In the last two rows, you might want to read a little more into this, is the fact that yes, the validation accuracy has a small improvement with the proposed method, which is what's desirable. But then going by what I said about not learning the background, the training accuracy reaches 100% much earlier compared to the other traditional models. At the end, I would like to say that this is this very simplistic experiment with really clean data. So not to read too much into these numbers as of now, but just indicative of the promise that this module provides. So yeah, stay tuned for more results from training data. And with that, I'll end my talk. Thank you.
So why don't we have time here for questions. I'm going to bring up the chat room. And if people have questions, why don't you type the questions in and I'll read them out so everybody can hear, and that way we won't have people overlapping. If anybody in this room has questions, let me know. I'm just gonna pause here for a second or two so we can see if there's anybody else that needs questions. And thank you. I have to say thank you very much for staying on time. That was perfectly timed.
Thank you.
And I just asked him how sensitive it was to input in terms of the quality of the sounds that got put in. Was it effective if you have real world data, or does it have to be really pretty training data in order to get those kind of results?
So the results that I showed here, as I said, it's trained with very clean data. So, that's not to be taken seriously. Mm-hmm. But yes, as I said in the earlier slides, it dumps down the background. So only your signal is highlighted for the system to learn. Mm-hmm. So yes, it has a promise to perform better in noisy conditions. Mm-hmm.
Okay, there's two questions that came in on the chat. How can you, three. See. How can you control in acoustic environments what the background is? Certain birds are found in certain acoustic locations.
So this is a deterministic approach. It doesn't really deal with specific species. All it does is if there's a signal that a human can pick out in a spectrogram visually, this module will help highlight that to the subsequent network. Did that answer the question?
You said no.
Maybe I didn't understand the question completely. Could you repeat the question, please?
Sure. Question is, we go back up. How can you control in acoustic environments what the background is?
Yeah, again, as I said,
so here's some more clarification. Isn't there a risk your model will learn the acoustic background if all the training data is from the same location?
No, so that it's the contrary actually. So this model does not learn the background at all. So, again, as I said, so anything that a human can perceive that stands out from the background, the system should be capable of perceiving those sounds as well. So that's what this module highlights.
Okay, and the last question before moving on to our next speaker is this system or this method used with only one microphone? What about an array to locate the sources?
This just does detection classification only. The localization is not included in this component.
Thank you so much for your time. I was muted there. We're going to need to move on to our next speaker, which is Michelle Fournet. Michelle received her doctorate from Oregon State University in wildlife science, where she investigated the impact of anthropogenic noise on marine mammals. She's an applied acoustic ecologist interested in human-wildlife interactions and practical applications of acoustics for marine conservation and restoration. Let me just clear my screen here. And Michelle, take it away. And I'm going to ask everybody else just a reminder to mute. All right, Michelle, it's yours.
Thank you. Can you hear me okay? Is it loud enough, or do I need to get closer to the speaker?
We have external speakers here, so I'll let somebody else answer.
I will assume I'm all right unless somebody gives me some sort of information to the contrary.
I heard it could be a little louder from UMass.
All right, how am I now? Is that better? I'm hugging my computer.
Yeah, I think that's good. Much better, they said.
All right, great. Thank you all for organizing this seminar. It's been a really cool series to be a part of. And it's really nice also to see kind of the breadth of the work that we as acousticians are doing. And I think that Shyam and I actually make a really good foil to one another, because we are on fundamentally opposite ends of the bioacoustic spectrum. And that from time to time I rely on Shyam's expertise for doing technical side of things, but really my interest is in learning how we can take sounds that exist in the natural world and make decisions. It's all about decision making and management and about conservation and restoration. So again, as Jen said, I'm an applied acoustician and an ecologist by nature. So I will have to watch Shyam's talk at least 10 or 20 more times before I think I can truly appreciate the gist of what's going on. And as an applied bioacoustician, I am interested in using sound to answer questions, but also questions that inform real world decision making. And in my time here at Cornell, I've been quite fortunate to spend time with evolutionary biologists and more traditional ethologists. And for me, it's constantly about bringing back what we understand about the natural world and using that. And traditionally for applied bioacousticians, we look at species that we're really interested in. So you can see that all of these pictures refer to projects that are done here at the Bioacoustics Research Program. So we have gibbon calls down here in the bottom. That's a spectrogram from my colleague Dina Klink. We have the Elephant Listening Project here, where we're trying to understand forest elephant conservation. And then we do work with impacts of noise on marine mammal species throughout the globe. And what is fairly common in marine bioacoustics and terrestrial and conservation acoustics in general is that we listen to species that we think need us, that we put microphones and hydrophones out into the world because there is something that we are lacking from an animal. We are concerned for its well-being. We are concerned for its function within an ecosystem. And we're trying to either do something to aid that animal, or we are trying to understand what the environmental impacts of an animal may be, and then we're trying to make management decisions. The work I'm going to present to you here is a little different. Most of this work is done with passive acoustic monitoring, and my work is done with passive acoustic monitoring too, and it is a big shift for me to move into fish. But you'll notice that my title said Passive Acoustic Monitoring and the Toadfish, an ecosystem-based management love story. Because contrary to a lot of the work that we as conservation acousticians do, I'm actually studying a species that is not at risk. It's prolific. It's vocal. It's not commercially important. From a human perspective, toadfish don't need us. And that is actually quite precious.
to study something that is interacting with its environment externally to being under conservation pressure or human extraction is actually exactly what makes this a good species for me to study from a management perspective. So let's talk a little bit about ecosystem-based management. Ecosystem-based management is a balancing act. And what we are attempting to balance is human activities, things that we need like water and land, and environmental stewardship, the right for a shark to swim in the ocean and a tree to grow on shore. And we're doing this across a multiple use context. So here you'll see we have a video that I took this past fall in Florida Bay. These are mangrove snapper, and these mangrove snapper are a commercially important fish. They are a recreational fish. They exist here in this particular ecosystem. Mangroves sequester an enormous amount of carbon, and this area is a habitat for lots of juvenile species. This area is also personally important to the residents of South Florida. And so there's a whole wide range of ecosystem services that are persisting right here in this video. There's money, there's health, there's ecology, there's climate change. If we attempt to manage any one of these things in isolation, then the balance of this ecosystem will fail. If we assume that this ecosystem should exist in the absence of humans and we should make it pristine, then ultimately what we end up with is a political battle, and we have a this or that mentality. But there has been a large move to shift away from the concept of wilderness or people to figure out a way to incorporate people and wilderness. How can we bring these two things together in order to maximize the services that, say, a mangrove forest provides to both the animals that live in it and also the humans that care about it? And my work is done in the Florida Everglades. And the Florida Everglades is colloquially referred to as a river of grass. This is Florida Bay. This is a photograph that I took just a few weeks ago, actually. Florida Bay is a large estuary at the very southern end of Florida with an average water depth of, I kid you not, 3 feet. You could walk across the entire thing if you didn't mind getting your boots wet and smelling a little bit like mud and muck. And traditionally, the Florida Everglades have been exposed to a very slow, steady downward flow of fresh water from Lake Okeechobee and the Kissimmee River down out across South Florida and then eking its way into Florida Bay. It's a very slight incline and it's extremely slow flowing fresh water. And because of this, things like temperature and salinity of the water in South Florida for a very long period of time remained highly consistent. And it would change based on the ebb and flood of things like storms and rainfall. But humans came along in the early 1900s and dramatically altered the water flow through South Florida. We had a need for agriculture. We needed fresh water so that we could drink and so that we could water our crops and take showers. And equally as important, farmers wanted to get that land out of the Everglades so that they could plant on it and so they could generate a food supply that was important for the settlers who moved into South Florida at the time. And so they quite literally drained the swamp. They rerouted the freshwater supply from Lake Okeechobee, and instead of having this slow, gradual freshwater supply to the southern regions, instead we have a series of dams and levees where we open up and let deluges of water out through specific pathways at certain times. As a result, we have dramatic changes in things like salinity and temperature and nutrient loading that make their way through the Florida Everglades, which terminates, as you can see down here at the bottom, into Florida Bay. And these changes have monumental impacts on the ecosystem. So you can see here on the left, we have a biologist that's doing a survey of a healthy grass bed. These grass beds are found throughout Florida Bay. They're very common. It's an important ecosystem for estuary fish. And further, these grass beds, seagrass, sequesters more carbon per hectare than a hectare of tropical rainforest. So it's a vitally important ecosystem on this planet, both for Florida Bay and also elsewhere. On the right, you can see this is a photograph that was taken in 2015 after a massive seagrass die-off. And that seagrass die-off has been attributed to dramatic and almost instantaneous changes in salinity. where we had hypersaline events as a result of changing the water flow to Florida Bay, and we have these ecosystem collapse moments. Seagrass is considered an ecosystem indicator for Florida Bay. At times when you have healthy seagrass, that's an indication that your ecosystem is healthy. Obviously, when you have the photograph on the right, things are not going to survive there with quite the same vigor as they will as the photograph on the left. But there is some issues with having seagrass as an indicator species. Is it scalable? Is this something that you can measure? And by the time you start to see changes in the seagrass, is it really one of those moments where you have to acknowledge that it's too little too late? Once the seagrass has collapsed, the ecosystem itself has collapsed. So while seagrass health is in fact a good indicator that Florida Bay is ecologically sustaining, seagrass death comes at a large toll, one that we are incapable of effectively predicting on the same scale that we can measure things like water quality. So as a manager, perhaps we need to consider other species. Let me introduce you to the toadfish. The toadfish, squatty and flat. The toadfish is not a specimen to be appreciated by looking at it, though still perfectly suited for its needs. That's a quote from one of the first descriptions written about toadfish in the late 1940s. And toadfish are a widespread species. They're found in estuaries. There's 79 species found throughout tropical and temperate regions. The Gulf toadfish is found throughout Florida Bay in our most marine environments as well as our most estuarine environments. And males are highly vocal. They produce something called a boat whistle call. They produce this during their breeding season in order to attract mates and to defend their territories. During the breeding season, male toadfish will nest in, if you look in the literature, they love to talk about the fact that toadfish nest in. If you throw a boot into the ocean, you're likely to catch a toadfish. If you throw a line into the ocean, it will likely ignore you. But they'll nest in any small rocky crevices inside of old tires. They will also nest near rocks, and often you'll find them in seagrass beds. They're not an endangered species. They're not commercially or recreationally fished. And importantly, they are a mid-trophic level fish. Sonic fish are an incredibly important food source for bottlenose dolphins, which we find in Florida Bay, for different shark species, as well as large barracudas, tarpon, large fish species that we find in this area. And they vocalize year round. So even though you will hear the highest number of vocalizations during their breeding season, you will hear toadfish calling during the nighttime hours and often during daytime hours all year. And this is extremely important because it means using passive acoustic monitoring that we can document toadfish behavior, presence, and absence at the same scale as the environment. So if we're taking things like water temperature on a minute-by-minute scale, then we can also take things like toadfish calling behavior at a minute-by-minute scale. And they are a model system for bioacoustic research. We understand both the anatomy and physiology of toadfish, the mechanisms for sound production in toadfish, and they can be investigated in the field or in the laboratory, which means we can go directly from understanding sound to understanding behavior to understanding ecology, and then for our purposes to see whether or not we can use this relationship to inform management for the sake of conservation and restoration. So for my work, I'm focusing here in Florida Bay. And there are 10 water quality monitoring stations that I do most of my work at. And you can see these here. And at these stations, we collect 24 hours a day in regularly scheduled intervals. We collect things like temperature, salinity. We do once monthly checks to get things like chlorophyll, turbidity, dissolved oxygen. So there's a number of long-term stations like these that calculate hydrology and water quality monitoring. 10 of them, the 10 that are on this map, have hydrophones that are hanging off the side of them that are recording as we speak. For this study, I looked specifically at these three: Buoy Key, Joe Bay, and Duck Key. And so I put recorders on them. It's a variation of the Cornell Lab of Ornithology Swift, which is traditionally a terrestrial recorder. We outfitted it for the marine environment, put a hydrophone on it, and they will record for up to 3 months at a time at either an 8 or 16 kilohertz sampling rate. And because the water is extremely shallow, they're only deployed down to 1.5 feet, I think is our maximum depth. And these are our 3 sampling locations. And we chose these locations because they're representative of a wide range of temperature and salinity regimes throughout Florida Bay. So on the far left at the top, we have Joe Bay. That is our most estuarine environment. And then we have Duck Key, which is getting slightly more marine, and then Buoy Key, which is considered a marine environment. And in this chart, what you can see is that we have tracked both salinity and temperature across the entire toadfish breeding season. Toadfish begin to breed in spring and they will end in late summer. If you can just barely see, there are 2 light colored gray lines, which is when I've actually focused my study, which is during the early March period, which is peak toadfish calling behavior. But in order to understand how environment informs toadfish calling behavior, which is my goal, it made more sense to look at these environmental parameters across a seasonal scale than at the minute-to-minute, day-by-day scale. Male toadfish have extremely high nest fidelity. When they select a nest, they're not likely to abandon it. So if I hear a toadfish calling in April, it's highly possible that toadfish selected that nest somewhere in February. So when I'm looking at differences in how toadfish act across these 3 different environments, it behooves me to understand something about the seasonality. And you can see in these plots that while the temperature changes quite a bit over the course of our season, it does change regularly between sites. Alternatively, salinity occurs across a gradient with a marine, a mid-estuarine, and estuarine conditions. So what I did for each of these locations was I listened for toadfish. I don't know if you'll hear this, but we will do our best. So those are a series of toadfish calls, the boat whistle calls that the males produce during their breeding season. And what I did is I subset our March data to periods just around sunrise and sunset. And I looked at the first 10 minutes of every hour directly prior to sunrise, following sunset, which are what we presume are these peak calling periods. For every 10-minute period, I annotated the presence or absence of a toadfish call in a 15-second time period. 15 seconds we determined was enough time to identify variation without getting into issues of multiple callers. If there is one caller in a 15-second time period, it generally will be non-overlapping with a second caller, and changes in those behaviors are closer to the call rates for toadfish. But because transmission loss changes between my different sites, counting calls is not actually a robust way to determine whether or not we have more or less in any one area. And then I looked at the presence absence of these calling male toadfish across 2 different scales. We scaled our data analysis. First, I looked at my between site variation, and I looked at the total observations across my breeding season during that period in March, and I quantified differences in temperature and salinity. So you can see here that my salty and saltier columns have slightly less toadfish calls than my not so salty column. In fact, there were more calling toadfish, or more, the probability of detecting a calling toadfish was significantly higher in Joe Bay, my least saline environment, than in my other 2 environments. And there was a statistically significant difference in salinity between my locations, but not so much in temperature. Alternatively, we also looked at the minute-by-minute probability of detecting a toadfish call. So we developed quasi-binomial regression models. These are generalized linear mixed effects models, and I used a drop-in deviance test for variable selection. And ultimately, I was testing the probability of a toadfish calling against temperature, salinity, moon cycle, and site. And then I included a random effective date and included various correlation coefficients just to make sure that we didn't have any significant temporal lags. And what we discovered, as these plots will show you, is that across our 3 locations in Florida Bay, the probability of detecting a toadfish decreased as salinity increased. And what was nice about these results is that this behavioral shift was predictable across sites. So the trend that we saw in an estuarine environment was the same as the trends that we saw in this marine environment. Within the threshold of where we were looking, salinity was negatively related to the probability of a calling toadfish. Now, there are 3 behavioral features that might explain this change in toadfish calling. Toadfish could abandon their nests. If it gets too salty in an area, a male toadfish might just up and leave. That's unlikely, but it's possible. Alternatively, our toadfish could cease breeding behaviors. If they're dealing with excessive environmental stress, then from a parsimony standpoint, that toadfish might stop calling because he needs to live to eat another day, and he can try again for the breeding season when the conditions get better. Lastly, changes in environmental parameters can indicate that mortality has occurred. There has been one example of hypersaline conditions within these 3 sites that resulted in a massive toadfish die-off. So while these are an extremely resilient fish, they are not impervious to the effects of hypersaline conditions. But this predictability, this between-site predictability, means that we can compare toadfish behavior across Florida Bay even when conditions are different. And importantly, that at 2 different scales, both regional and locally, that our toadfish are responding to changes in their environment. So does that make them a good indicator species? Does that mean by listening to toadfish that we can make informed decisions about management that will tell us something not just about what toadfish are doing, but will give us information about Florida Bay in general? The goal of a successful estuary indicator is to evaluate overarching conditions without having to dig into the full complexity of the system. That's why we look at species like toadfish or seagrass, or in some cases, snapping shrimp, is because we like to think that rather than tearing everything apart, that we can look at something which is interacting with its environment and learn something about that environment. But to be a good indicator species, the animal needs to be broadly distributed, which toadfish are. It has to have limited movement between sites. As I mentioned, male toadfish have really high nest fidelity. They'll pick a nest and they're going to stay there for the breeding season, which means that the toadfish that I'm listening to in Joe Bay is not the same individual that I'm listening to in Bowie Key. These animals need to be biologically responsive to their environments. They have to give me some measurable indication of what's going on in their system. And then similarly, they need to be ecologically important. Now, in this study, I can demonstrate that they're broadly distributed, that they've limited movement, and they're biologically responsive. But this food web that we have on the other side of the screen is something that we still need to completely unpack. Are toadfish, in fact, using acoustics in order to interact with each other? And does that interaction end up calling in predators? Are predators cueing into the sound of our breeding toadfish? Are prey running from the sound of our breeding toadfish? And importantly, theoretically, if we removed toadfish from the equation, are we going to have a measurable change in the ecology that then is manifested through these studies? Are the dolphins going to suffer? Are the crabs going to overpopulate? If these things are true, then knowing what's going on in toadfish ecology gives us a clue as to what's going on into the broader ecology of Florida Bay. This is also important because it is feasible. I can partner with Shyam. He can help me develop a detector for my very simple toadfish call. We can then use this to look across Florida Bay at meaningful, environmentally relevant timescales so that we can assess changes in our ecosystem. I can do this without ever catching a fish. There's something important about that, about developing scalable and relevant metrics that have low impact to their environments. It's been said that the Everglades is a test, and if we pass, we may get to keep the planet. This is an ecosystem that we, with our human needs, have dramatically altered. But it's also an excellent opportunity to demonstrate that ecosystem-based management can work. That if we spend some time listening under the water, looking above the shores, and figuring out what our priorities are, we can perhaps solve what we once thought was an unsolvable problem. So that's what I've got for you, and thank you very much. And everybody should make their way down to Florida Bay to go and take a kayak trip through because it's just spectacular.
Yay, thank you, Michelle. Okay, we've got clapping on our chat here, and we're going to do the same thing. So if you have questions, I'll read them out, and here comes some. They come already. Very nice talk. Love the way you zoom in from a big ecological question to your specific study. Your plot of probability of calling versus salinity looks like it has a convex curvature. Does that fit with your result saying there is a consistent delta probability per delta salinity?
Yes, that's absolutely true. And at some point, actually, if we had salinity values that were higher, we would probably end up seeing a quadratic, that at some point we're going to fall off entirely and our fish are either going to die and therefore we're not going to get anything. But at higher levels, there does seem to be a more dramatic impact of salinity.
Awesome, that was a good question. Any other? And I have to say, these two talks were very complementary. I really liked the dichotomy, but the complementary also between them. You guys are very fortunate to be working in the same lab.
Yes, absolutely, 100%.
Okay, do we have any other questions? I just like to pause a little bit here and kind of fill the silence to give people a chance to write in. Oh, here's another one. What is the scalability of this in terms of to other regions and sites?
Oh man, that's the most exciting part. It's extremely scalable. These fish are broadly distributed, and there's 9 different species of toadfish that are really biologically very similar. They fill similar ecological niches, they share behavioral traits. And so one of our goals actually is to see whether or not we can demonstrate that toadfish might be a good either indicator species or sentinel species across extremely broad scales. So you'll find these in tropical environments. The splendid toadfish, everyone should Google it, it's just beautiful. Also produce vocalizations and are extremely resilient species, but also responsive to their environments. And so one of the things that we hope to do next is narrow down which sites in Florida Bay are the best sites for doing toadfish monitoring, and then start looking in areas like Biscayne or possibly throughout the Gulf of Mexico, where they have either the same Apsanus beta species or similar species that we could use, again, to understand things about the environment. And then because there are species that you can keep in captivity, if you have flow-through tanks, there are ways of experimentally gauging their responses to the environment and finding out where their thresholds are. So if we could experimentally assess what their thresholds for behavior shifts were, then we could potentially start looking at toadfish as indicators of environmental parameters, even in the absence of things like really long water quality monitoring stations. So this species is a particularly good species for scaling both throughout the subtropics and also into temperate environments as well.
Okay, we have a question here in the room.
Hi, Michelle. This is Tony Lyons at UNH. You kind of quickly went over, you stated a sentence that these 3 sites had different propagation conditions. And are you sure that that doesn't have an effect on your measurements? Or did I hear that it didn't have an effect?
At the scale that we were looking, we don't think it has an effect. And that's actually one of the things that we have done and that we will continue to do is actually drop a speaker in the water to measure sound propagation in this area. They were all placed at approximately the same depth, but there is differences in the bottom substrate. So some of these have muddy bottom substrate, some of these have sandy bottom substrate, and some are closer or further away from mangrove forests. So the reason that we aggregated our data instead of counting calls was exactly for that reason. So there were definitely times in the recordings when I could hear lots of really loud calls at one site, and then at another site the calls were much quieter. So I can't necessarily control for propagation in terms of how far my listening range of my hydrophone is. So instead we tried to account for that quantitatively by designing a study that would look at it across an ecologically meaningful scale. But stay tuned, because we're headed back down with a playback speaker and a whole suite of tones and upsweeps and also toadfish calls so that we can empirically measure what the propagation is for this region.
I've got 2 more questions here. How might you distinguish the potential toadfish behavioral responses? Reduced mating, leaving, or death?
Well, for that, what we're hoping to do is to start looking at some census data. So if we can get trawl surveys out there, and we do have colleagues that do specimen collection in that area, then we could see whether or not we actually have reductions in populations versus just behavioral shifts. Death is actually pretty easy to quantify because people are out there all the time. So whenever there's massive die-offs, the fish just float right to the surface and it makes a big icky mess and it gets reported to us quite quickly. But looking at things like nest abandonment versus looking at changes in behavior, that one's a little bit harder. One of the things that we have toyed with the idea of doing is actually going through and putting out traps and doing things. There's a known growth curve for toadfish, so we could actually get age as a function of size for these animals. So looking at recruitment across these different areas to see if there's preferential recruitment. And then always the goal would be to couple any field effort we do with experimental design to see whether or not if we control changes in salinity, do we see a change in behavior versus toadfish that might actually leave the substrate that they've been protecting.
Okay, last question, and then we'll be out of time, is what is the, I think, frequency of these calls? The spectrogram did not have units. they also had Tony's same question about propagation. So propagation seems to be of high interest here. But just what was the frequency? Because that would be key to understanding propagation.
It's fairly low frequency. I want to say 100, 200 hertz, maybe 200, 300 hertz. Hold on one second. I can.
Yeah, that's about right. That's typically what I've seen.
Yeah, 200, 300 hertz. And now, because this is such a shallow water area, low frequency sound doesn't actually propagate very well here. Actually, Jen, I think it was your work. I was reading about manatees and how manatee sounds propagate through these shallow water environments and higher frequency for such a large body size. So one of the running theories as to why toadfish are using low frequency calls in a sound environment that doesn't favor that is actually because it reduces their communication space and limits predator accessibility so that they're not advertising to predators. And because they nest in one area and they nest in somewhat high densities, female toadfish presumably are coming into these nesting regions, areas that are also good spawning regions for them, and are in closer range to the male toadfish. But these sounds don't travel that far. And so for our purposes, I don't know yet what the difference in communication space is for a toadfish in one of my environments versus the other one, but it is 100% on the table to find out.
One of the things too is particle motion. These fish are sensitive to particle motion as manatees are, or vibration in the substrate. And so that's another aspect that can be considered.
100%. If anybody is a specialist in that and wants to set up some time to chat with me, I would love to pick your brain about ways that we might be able to quantify that in the species, either out in the field or in the lab.
Okay. Well, thank you very much to both of our speakers, and I'm going to conclude this for today. We're out of time, but hope to have see everybody back or logged on in April. All right. Thank you so much.
Thank you. Cheers. Bye-bye.
Have a great day, everybody. I know. I have to stop recording.